Hello, good afternoon, everyone. Viewers at home, this is another opportunity to learn at the feet of Olatunde Michael under the educational series of Omega Bio-Edutainment. Today is going to be another follow-up and furtherance lecture on DNA. The last video we had, we actually look at what are the molecular basis of mutation, and we also consider what are the biological effects of mutation, whereby we were able to look at what are the molecular scenario where we have mutation. We actually started with point mutation that involved substitution. That is when you have removal of a nucleotide base, and you also have replacing it with another nucleotide base. And we actually look at that kind of a scenario. After that, we look at a frame shift, which actually involve insertion of an extra nucleotide, and we were able to look at what could be the effect on the that kind of a protein. Then again, we also look at a deletion mutation, which has to do with removal of a nucleotide, which actually leads to a shift in the reading frame. Then we went as far as also considering that if this mutation will not actually take place at the DNA level, what of if it is occurring at a chromosomal level? And we cited an example of a chromosomal mutation where you have duplication of certain gene on the chromosome, and we were able to look at this, that is going to lead to overproduction of certain protein, which is an example of a tumor, that is a cancer. And the last thing we consider was a kind of translocation mutation, which will actually involve exchange of a chromosomal segment that has to do with two different chromosomes having gene A and gene B being exchanged. And we cited an example of what happened in human chromosome 9 and chromosome 22. That was a basic example, or a typical example of where we have a translocation mutation, and which resulted in a disease condition which is known as a chronic myelogenous leukemia. So that is another form of cancer. So today again, the problem, you should also begin to ask yourself, and I told you, I've explained the reason why our focus has only been on DNA repair, DNA damage, what is going to happen. And that is going to form the basis of our discussion today. So, and that is why we are considering mechanism of DNA damage, as well as what is a mechanism of DNA repair. Okay. Just like I said, everything that has to do is actually bordering on DNA, and you know that talking about DNA is the effects or our over concentration of DNA cannot be overemphasized, and that is actually one of the reasons why your primary, as a biologist, whatever type of biology you could be, you should be able to have it on your fingertips. The structure of DNA, what are the composition of DNA, and what are the processes by which DNA perform its normal function, and even how important DNA could be if there is an error. The slightest error to DNA can just go on and manifest in different, I mean, let me say in several generations. So that is the reason why you have to actually talk about all this. Okay. So just like I said that we ought to know why our interest is going to be on DNA. So I have told you that DNA is very, very important, and emphasis on it should not be over taken to be something that is not actually that. So today we want to look at DNA again. We want to look at DNA damage and how is a DNA going to repair that damage. Every process in life has its own risk. It has its own hazard. Every facet of life, every aspect of life, every profession has the benefits as well as the risk that is actually going along with it. So you see, DNA damage, the first thing is we want to look at the damage. What are the likelihood, what are the possibilities of DNA undergoing what is called damage? Don't forget that at this point in time, I cited an example of the normal processes in which DNA is being involved. So without DNA, there cannot be anything that is going to be transferred from their parents to the offspring. That's why they call DNA the hereditary material. So the only thing that you will carry to any other is going to be that to the any other generation is going to be that you will have to be very, very sure that your DNA is performing its normal function and duties. Now, so DNA damage, the damage or the risk by which DNA is being exposed to can be as a result of two factors. It can be internal. The damage can be internal, that is within the DNA or within the cell. So, and it can also be what? External. So, it can also be external. If it is internal, it is called endogenous factors. What are the endogenous factors that affect that causes a DNA damage? Then if it is external, it is called exogenous factor, that is, it is outside that. And if there is no damage, there won't be anything that we call repair. If your car does not break down, but don't forget that even a car will actually need that from time to time. You will need to put the car in shape. It's not going to be until the car has broken down before you'll be able to know that you need to do what is called maintenance. And yesterday, I think I cited an example of some of the proteins that are regularly on routine basis that they will be moving around immediately after the DNA replication. They will be moving around to actually conduct a kind of check at every point in time. I told you that they will have the normal one, that is the normal DNA. They will have it, and that is what they will use as blueprint to be able to control what is going to be the type of repair that they see. So then, not only that, these protein, some of them also have the capability of fixing the error, of repairing the DNA. So, and that is exactly what we want to talk about today. But something should be rest assured in your mind that every kind of damage that is natural, that is normal, that is spontaneous, every kind of damage, the nature has actually empowered each cell to be able to repair its error. So if everybody is as even as an organism or as human being, we are prone to make errors, and under normal circumstances the error that we are going to commit should not be error that will be beyond repair. So the same thing with DNA. So it has its own mechanism of repairing all the damage that could actually call for. In fact, DNA always spend more energy. They have more energy to fix up errors than even the error that they will use in making the normal replication. And the reason is very best understood that if you don't have money to maintain your car, why do you actually have to buy the car even in the first instance? So, and the money you always spend on maintenance, if you combine it together, that is, if you add it together, that money is going to be times in multiples of what we are going to use in even repairing it. So the repairing mechanism, so by the time we are understanding, by the time we are going through what the damage could be, we also need to understand. In fact, under normal circumstances, it's just because of our time, although I'm going to also dedicate some video that is going to treat replication. So we are going to look at replication. So this one is the normal process by which every DNA must be capable of doing, because if there is nothing, if the DNA will not have to undergo replication, that means there's nothing that is going to call a kind of growth. Development will not be there. So, but as much as this one is a normal, so it is normal for us to be able to work every day as human being. You must work, but by the time you are working, you will make mistakes. By the time you are making mistake, you should be able to have the problem. I mean, you are able to have the mechanism that you are going to make it repair. Don't forget that even right away from primary school level, that you don't even know what to write. As soon as they are teaching you how to write alphabet, you are going to be at that point in time, they will also teach you that you need eraser, you need cleaner to be able to clean whatever that you know is going to be a mistake. So that is actually telling you that if there is any process and that process does not in any way make way for mistake and also make way for it to be that mistake to be corrected, you will see that that is not a correct kind of mechanism. So what we are going to start with, as I told you, I'm going to dedicate one or two videos that will make us to be able to understand the process of DNA replication. How do DNA replicate, and what are the basic mechanisms that you must know, because it is a technical term that has to involve the two strands of that DNA. Okay, the way I told you there is going to be antiparallelism. One strand of that DNA will proceed from 5 to 3 prime, and in obeying the law of antiparallelism, the other strand is going to start from 3 prime to 5 prime. So the way this one will undergo replication will be quite different from the way this one is going to undergo replication, and that's why you have what is called a lagging strand and leading strand. We are going to see all this when we consider what are the mechanism of DNA replication. So we will first of all start with the damage, that is errors. DNA is prone to errors, just like human beings. We are all prone to error, and the way we repair our mistake, the way we repair our error will actually determine whether we are going to be a successful person or not. Don't forget that yesterday I cited an example. I mean, one of the primary, let me say, the primary truths. Let me just use that. The primary truths about DNA is that the integrity of the DNA must be conserved. The fidelity of that DNA must be conserved. So all the composition of the DNA, the one that starts the process should be the one that is going to also complete it, and is going to be this. So it's just like at the end of the day, okay, so let's consider the mechanism of DNA damage. The mechanism by which DNA can be prone, or we can have problem with DNA. So there are various factors. And these factors will target the structure of that DNA. And the first damage that every DNA is prone to is the physical damage. So physical damage to DNA. How do DNA damage itself? How do they damage itself with physical damage? And when we talk of physical damage, these are kind of damage. So damages or errors due to one, ultraviolet rays, that's a ultraviolet light, okay, e.g. especially the one that is caused by UV radiation. You should know that every day we are being prone to what? This kind of damage. So because we will continue to expose ourselves to the sun rays, and and when we expose ourselves to sun rays, this one is prone to damage our DNA. So how would this actually happen, and what are the examples of this, and what would be the cause? What damage would they cause? Number one is that there will be formation whenever we are being exposed to UV light or UV radiation. So it's going to cause us a kind of it's going to lead to error that is due to pyrimidine or formation of dimers. Especially thiamine. Okay, thiamine dimers. So this kind of error is when thiamine damage. So this happen when the two adjacent thiamine bases covalently bond with each other. Covalently bond and thereby distorts, and thereby is going to distort the DNA helix. Okay, it's going to distort the DNA helix and causes replication error, and thereby causing what is called a replication error. So that is the first thing that has to do with involvement of damage. Okay, don't forget, for example, this is our DNA, double-stranded. Okay, now if you now have T, you have T, you have A, you have G, for example now. So this one is going to be law of complementarity will tell you that this one will be A, this one is going to be A, this is going to be T, okay, this is going to be, okay, let's say this is C, this is going to be G, then this also is going to be C as well. This will be double bond, double bond, double bond. Now, under normal circumstance, if you should remember very well, the double bond, even in chemistry, they are prone to form what is called dimer when two double bonds they interact with each other. So this double bond, because it is double bond, they are prone to what is called dimerism. They will undergo a process called dimerism. That is, you have two die from two, dimerism. Okay. Now, whenever there is now dimerism, when you have thymine, thymine and thymine being covalently bonded to each other, covalently bonding to each other. So, whenever this one is now formed, if this one will now undergo replication, it's going to replicate itself. Okay. So, replication, it means that this strand is going to produce another strand that's going to be like that. Okay, so going to be like that. So during the process of this is replication, during the process of replication, because a dimer has actually, this is a dimer, or dimerism has taken place between this adjacent thymine, so it's going to leave a gap during the process of replication. It's going to leave a kind of gap in this process, and that is an error that is as a result of damage by ultraviolet light. So this one itself, dimerism is an error that is due to UV light, ultraviolet radiation, when the DNA becomes so exposed to sunlight or X-ray or wherever, that there can be a source of irradiation to take place. The error that they will form is going to be error of covalent bonding that will take place between two adjacent bases which are covalently bonded together. So thereby is going to distort the helix. Once the structure, I told you, you have to let this be an established fact. Once the structure of anything, especially in this perspective, we are talking of DNA protein. If the structure is affected, definitely the functionality is also going to be affected. And once the structure of the DNA is already been disturbed, you know what is going to cause now. For example, let us assume that you know what is going to cause. So we might not actually need to still make a further assumption. So, and during the process of replication, it's going to distort the helix and thereby causing the replication error. Causing replication errors. Okay. So don't forget that immediately after this lecture, you are to actually form a kind of a summary that to summarize this lecture in not more than few sentences. Not more than few sentences, like that will not be more than two that will not be more than 300 words. You have to summarize this that will not be more than 300 words. Okay. Now, that is the one that is due to UV light. The B, that is under physical damage, is the one that is due to ionizing radiation. Ionizing radiation. And when we talk of ionizing radiation, we are actually referring to X-rays, okay, and gamma rays. So X-rays and gamma rays. Some of us might be wondering, how are we going to actually make sure that, how are we going to expose ourselves to radiation, X-ray? I'm not sick, and I've not undergone, I'm not going to subject myself to anything that's going to involve X-ray. No. Once you are consuming, there are some foods, synthetic food in particular, that they use ionizing radiation in order to prevent the growth of microorganisms on them, especially tin food, canned food, sorry. So all this one, the primary aim was to actually make use of this radiation to be able to prevent the growth of microorganisms in them. However, they cause a lot of damage to our DNA. And that's one of the reasons why they will always tell you to stay away from synthetic food, because it is a source of physical damage to our DNA. And don't forget that whatever happens to our DNA, I've been telling us that it is going to have a lasting effect. So, and whenever our DNA is exposed to this, it's going to cause what is called, it's going to cause what is called double strand breaks. It's going to cause double strand breaks, and that is DSBs. Double strand break. Okay, and sometimes it will also cause single strand base as well, breaks, also SSBs. Okay, we are going to see how all this will actually take place. And whenever there is either double DSB or SSB, it will eventually lead to production or creating, it will eventually lead to creating free radicals. Some of us that have undergone and are still undergoing toxicology, you will understand what is going to be the eventuality, or how creating free radicals, it will eventually lead to creating free radicals, and free radicals that is so toxic to our DNA. That is so toxic to our DNA. If you have bad DNA, you are going to have bad protein. You are going to have bad enzymes at the end of the day. So that is how what we can say about how our DNA is so prone to errors that is caused by physical damage. An example of that is UV light, such as ultraviolet radiation that you have from sunlight. And the first one is pyrimidine dimers. This happens when the two adjacent bases of thiamine in particular covalently bonds together, and thereby distorts the helix structure of DNA, thereby going to lead to causing what is called replication. replication error. And the second one is we look at ionizing radiation. For example, they use X-ray radiation and gamma ray in can and food industry very, very well to be able to sterilize the some of these canned food. And in thereby, by the time you now consume all those synthetic foods that are canned, you are prone to having double strand breaks or single strand breaks. And this will eventually go on by releasing free radicals that is going to be too toxic to our DNA. Okay, and some of the chemicals in this category is what is called, so they will release toxic free radicals of OH, that is hydroxyl ion. So this hydroxyl ion, the hydroxyl ion will interact, they will normally interact with DNA. They interact with DNA, and when they interact with DNA, they will form what is called 8-oxoG. They will react with guanine. They always interact with guanine G in the DNA. The first one will interact with thiamine, which is a pyrimidine base. Now, the second one, which has to do with ionizing radiation, will interact with guanine, and by the time it interacts with guanine, it's going to form what is called 8-oxo-G. And by the time you now form 8-oxo-G, that is going to cause a serious error. It's going to cause a serious error, and this 8-oxo-G is a known mutagen. So it's a mutagenic compound or mutagenic substance. That is, it can cause mutation. And don't forget that mutation is the beginning, just like I said, is the beginning of cancer. So these are the two areas whereby there is, our DNA is so prone to damage by physical damage due to ultraviolet rays, as well as ionizing agents like our X-ray as well as our gamma ray, which they use in preserving some of the canned food products that is being consumed by us. So this one is going to lead to single strand breaks as well as double strand breaks, and it will eventually lead to creating free radicals thereby. And I told you that in toxicology, even in advanced toxicology, we know the implication of having any error that is due to producing free radicals to DNA. So all these free radicals, they are toxic to our DNA. They will form compound that is going to be so toxic to the DNA that is going to lead to causing a very serious effect, having a very serious effect on the overall integrity and fidelity of DNA, which is very, very important. So we are going to look at the second type of damage. We are going to look at the second type of damage. Don't forget, the first type of damage is a physical, and we have told you what could be the source, the origin. How do we come about physical damage to the DNA? And we have seen that the first one could be from ultraviolet rays that we normally have from getting our DNA or ourselves being exposed to ultraviolet rays. So that is, and ultraviolet rays is going to lead to what is called pyrimidine dimers, and this pyrimidine dimers is going to is a kind of covalent bonding between two compound of thiamine, because thiamine is going to be double bonded to this. It will undergo what is called dimerization, and that dimerization is going to distort the DNA helix and it's going to cause replication error. So, and I've told us about the second type or the second sources of DNA damage, which is due to gamma rays and X-ray. These gamma rays is what they normally use in preserving our most of our synthetic food, especially in canned industries, like you see in canned industry. And this error is going to lead to production of double-stranded strand breaks, as well as single-strand breaks in DNA. And you know what it's going to cause. It's going to lead to production of free radicals that is going to be too toxic to the DNA, thereby forming an example of what is known as 8-OSO-G, which is a known mutagen. And a known mutagen or mutation is just the primary, the foundation, the beginning of cancer. Okay, so we are going to look at the second has to do with the chemical damage that is due to certain chemicals. Okay, certain chemicals. So certain chemicals, there are chemicals. This is going to be number 2. Chemical damage. Because the first one has to do with a damage that is caused, that caused the DNA is a kind of structural damage, physical damage that you can see. When dimers is formed, you will see it there. It's something you can see on that DNA. And whenever even there is the second one that is also formed, that is when you have OXO-G being formed, that is also very, very visible that you can see. So the next one is a chemical damage. So, and under chemical damage, the first category has to be the damage that is caused by alkylating agents. Alkylating agents. Okay. So this is formed. This alkylating agent is all these methyl giving methyl, all these all the compounds that contain methyl groups, such as methylbenzene. For example, now if you have a benzene like this, and you have CH3, this is, you don't need anybody to actually tell you. All the compounds that are methyl yielding compounds, they are under the group. They will be classified as what? As alkylating agent. Not only that, methyl group or ethyl group type. So even if you have something like this, there are some compound that they will be like that, C2H5. All these are grouped under alkylating agents. So these are alkylating. Okay, you need to know that. So, and the type of damage that they will cause is going to the DNA is going to be chemical in nature. So they will cause what is called mispairing error. They will cause mispairing. Don't forget that all the nucleotide bases of DNA, they also have a methyl group. Some of them have methyl group, that is going to, so it's going to lead to mispairing of bases, mispairing of bases during replication, going to lead to mispairing of bases during replication, and that can only be caused by alkylating agents. Okay, then the other one, we have the oxidative stress. Oxidative stress. Okay, just like what I said in the other type too. What I said in the other types are oxidative stress, which is as a result of there's something they call ROS. That is a reactive oxygen stress. All those that are in environmental biology, they will know much about ROS because it's a known toxicant. It's a known toxicant. There are some food that's going to lead to production, release of ROS. And when you say reactive oxygen stress, that means anything that's going to react with oxygen, like to leave us with very poisonous ozone, or it's going to lead to production of peroxide. All this one that will give us OH and all the rest. So all these, they are known as a reactive oxygen stress compound. Okay, and this one is also going to lead to production. It's very similar. It's going to lead to production of 8-oxo-G. Going to lead to production of 8-oxo-G. And what will oxo-G do? Oxo-G will eventually mispair. It's going to eventually mispair with adenine. It's going to mispair with adenine. Instead of thymine to pair with adenine, it is as a result of reactive oxygen stress that's now going to form this 8-oxo-G. And when this one is formed, it's going to react with adenine in the DNA. It's going to react with adenine in that DNA, and by the time this one is actually interact with adenine normally, because guanine is supposed to bond with C. So because 8-oxo-G has been formed, instead of it to react with C now, it's going to now bond with A instead of T to bond with A. So, and thereby it is going to lead to free release. Going to lead to release of free radicals. And all these free radicals, they are known toxicants. As well as known mutagens. Okay, so it's going to be very, very toxic to that. Then the C aspect of the chemicals are the chemicals that will intercalate. So we call them intercalating agents. So from the word intercalate means something that is going to wrap up, going to cover, going to form. Okay, so intercalating agents, and these intercalating agents, what do? They are going to insert themselves. They insert themselves into the DNA. They will insert themselves into the DNA. They wrap themselves into DNA and leading to frame shift. You now know what is a frame shift mutation now, leading to frame shift mutation. There are lots of intercalating agents, lot of examples on that are called intercalating agents. So although we are going to actually devote some time to learn about giving them examples, but basically the example in this case is what we have actually made mention. We have mentioned it. We have mentioned it like a glycolase, glycosylase. Okay, so all these are kind of intercalated. So it's just like when you have your normal DNA, which is a double-stranded, So you now have C, you now have T. You have G, you have A, you have T. So what you now have as intercalating will now be that it is going to certain chemicals. There are certain chemicals that they have the ability to cause a kind of frame shift mutation. They will go and replace. They will take the place of the normal nucleotide bases. They will intercalate. They will go and cover up. And by the time they cover up, they are going to form a kind of there's something they will form. We call it kink. They will form kink structure, K-leg. They form kink structure. So it's going to lead to error that's going to affect the structure of that DNA. And when the structure of DNA is affected, that means it will be very difficult to undergo the normal process of replication. So that is an error that is as a result of a kind of chemical mistake that DNA can actually make. We cited the example of the first one we consider was all the compound, especially the aromatic compounds that are methyl or ethyl yielding aromatic compound. They are better classified as alkylating agents, and when they pair with DNA, they will form a kind of mispairing of bases, and we know what a mispairing of bases is going to cause on DNA replication. So like that. And the second category is oxidative stress, like a reactive production of a reactive oxidation reactive oxygen stress. So we look at some chemical compound that will lead to production of 8-oxo-G, and 8-oxo-G, instead of the guanine to be able to pair with cytosine in the normal base pairing rule, is going to base pair with adenine, and thereby is going to release certain free radicals, and these radicals, they are known to be good toxicants. They are known to be good toxicants as well as mutagens. Okay, so apart from that, the third category of chemicals that can cause chemical damage are the group that they call them intercalating agents. They are intercalating agents. They only perform their own hazard or havoc when they form themselves, they wrap themselves on DNA. They just wrap themselves on DNA, thereby forming a kind of or leading to frame shift mutation. So, and we also know what is going to be that. Now, the third What's going to be the third types of damage that we can witness is a damage that is going to be replication damage. That the error will not be coming from either physical damage or is going to come from damage as a result of certain chemicals. Okay, but it's going to be the error that is going to involve replication. Don't forget how important replication is in maintaining the integrity of DNA, maintaining the integrity and fidelity of DNA. There is a difference between integrity and fidelity of DNA. So I want you to explain that as an assignment. What do you understand? What do you understand by fidelity and integrity of DNA? Okay, the fidelity and integrity of DNA. So we have to give hypothetical example. We have to give hypothetical examples in each case. We have to give hypothetical example in each case. Okay, and that assignment is actually due. Now I've given you two tasks now. The first task is to be able to summarize all my lectures today. Just summarize it in not more than 300 words. And the second task is going to be, what do you understand by fidelity as well as the integrity of DNA? What do you understand by it? Then you are also to give hypothetical examples of to explain fidelity of DNA as well as also explain the integrity of DNA. Okay, I'm also going to give you the third task before the end of this lecture. So let's consider replication error. Error that is due to the normal way of life of DNA. So the normal thing, the normal procedure, the normal activities is that every DNA must be able to undergo replication. I have mentioned it several times, even in this lecture, that about replication of DNA. So the first one, so that means after the normal way of life of DNA, so DNA is also very, very subjective to making error. Okay, so the error that we notice, the first one is called mispairing. Mispairing, error of mispairing. So, error of mispairing. There are sometimes during the lifetime, every DNA is prone to make a wrong choice in marriage. The same thing that we have it in human. Okay, that is error of mispairing. And you know what? Error of marrying somebody who is not even compatible with you. So the same thing like that. So mispairing, error of mispairing, that is when DNA incorporates, when DNA itself, it is the DNA incorporates wrong or introduce wrong pairing, long pairing of nucleotide bases, and sometimes it might not even be nucleotide bases. Okay, so and this one is due to error in DNA polymerase that is made by an enzyme called DNA polymerase, because this DNA polymerase perform a very vital role in replication. So instead of if the DNA polymerase is now malfunctioning, if it is malfunctioning now, you're just going to match somebody who is not even supposed to be your partner. Wrong pairing, wrong incorporation of a nucleotide basis, which is going to lead to what? It's going to lead to which may or may not, which may lead to error of substitution. By now, you know what is error by substitution, okay, if it is not corrected. Then the second thing during replication, the second thing that DNA is also prone to is the error of insertion and deletion. This error is very, very common. The error of insertion, that is incorporating a wrong base, or it could be a new base, nucleotide base, instead of it. You see, it's just like somebody who is having his car, maybe after the normal thing, instead of the person to service, you now give your car to a mechanic that is going to service your car or going to fix a car for you. So instead of the person to change the plug for you, the person now say, why do you want to waste a plug? Instead of 4 plugs, let us remove one and let's put 3 plugs. So will the 3 plugs serve the same function as to move your car as 3 we do? I mean, as 4 we do? No, definitely no. That is error as a result of deletion. And instead of it to be 4, you know, let's create another space for 5 instead of it. So it's still going to be there. So that is the same way that error of deletion as well as insertion could do. And insertion and deletion can lead to a frame shift. Can lead to frame shift mutation, which can be synonymous to death sometimes, or it can lead to frame shift. It can also lead to repetitive, or what is called duplication. Just like you know what duplication could do. Duplication of genes or nucleotide. So instead of you to have CGT, you now have CGT. For those of us in genetics, in environmental biology, that is in genetics, you will know what the error of having repeated CAG, what it can cause. CAG, you know what it can cause now. So it can cause what is called Leber optic neuropathy. Okay, that is one genetic disorder that is as a result of CAG repetitive this in the mitochondria. So the same way, if there is an insertion, it can lead to repetitive, that is, or duplication of certain genes. Okay, so that is that about the error that is as a result of a replication error. Okay, so finally today, the task 3 today is that you should list seven common mutagens and carcinogens. List them that are very common in your area. List them. The 7 common mutagens and carcinogens that can cause DNA damage. That can cause DNA damage and suggests the mechanism of its repair. You have to list 7 common mutagens and carcinogens, okay, and you have to suggest the mechanism by which the error that is caused by this carcinogen and mutagen suggest the mechanism by which it can be corrected. Okay, so lastly, the last category is endogenous sources of damage. You know, I told you that sources of damage before we started this class. I told us that there are 2 factors that can cause DNA damage, and the fourth one can be as a result of exogenous cause and endogenous source. So all the other ones that we have considered, physical, chemical, replication, all those ones are exogenous. So let's look at what could be what is called endogenous sources. So, and the first one we are going to look at is the one that is due to spontaneous hydrolysis. So the one that has to do with water producing reactions, and it's going to form a kind of hair. So under normal circumstance, you should know that the effect of water on DNA, the effect of water on DNA will cause what is called depurination. That is removal of purine. Depurination. Okay. Or it can also lead to what is called deamination. That is removal of amine groups, of amino acids. Depurination and deamination. So depurination is when you have loss of purine, and deamination is when you have loss of cytosine and uracil. So loss of cytosine and uracil is as a result of spontaneous hydrolysis. That is when there is a sudden generation of water molecules with DNA. It's going to lead to the error that is going to result into loss of purine or loss of cytosine or uracil. Okay. Then the second and the last thing that can also happen is DNA replication stress. The one endogenous replication stress. So just like when you can also overwork yourself, the same way that DNA replication can also overwork itself. So DNA replication stress can lead to production of of incorrect replication fork. Okay. Then secondly, it can also lead to generating breaks. There will just be break. Can also lead to break. That is, there will just be gap, production of gaps like this. So you might not actually appreciate what breaks are until we treat replication fork. When we have a generation of Okazaki fragments. Okazaki fragments are these all small fragments. Small fragments is after that ligase will now continue to merge all these these fragments together. So, like breaks now, under normal circumstances, the leading strand is supposed not to generate Okazaki fragments. It is only the lagging strand that's supposed to generate Okazaki fragments. If on the leading strand you now have generation of gaps, it will lead to breaks, and this is a result of a DNA replication stress. Because you see, whenever any system overwork itself, it can lead to so many errors. Just like you, that you overwork yourself from morning to night, you don't have time to actually rest. I told you yesterday that after undergoing replication, after undergoing cell cycle, prophase, metaphase, anaphase, and telophase, every cell will undergo a process or a phase where the cell will rest, the cell will rejuvenate, even if it might not be more than 1 second. Sometimes when you overwork yourself, you just need few seconds to actually rest and relax so that you will not make errors. So the same thing, this kind of error of what is called DNA replication stress also is an endogenous sources. Endogenous sources which can lead to production of incomplete replication fork. You just be making mistake when you do not rest, and they ask you to even just calculate add 2 plus 2. You might say 2 plus 2 is equal to 6. It's going to generate a lot of errors. So that is why the system has actually developed certain mechanism to be able to repair all these errors that can be it endogenous or exogenous. The cell is very capable of doing that, and that is why that's going to be the focus of our discussion today. The next time we will come, we will now understand what is called what are the mechanisms that the cell must put in place in order to be able to repair all this error. Don't forget that at the beginning of this lecture, I told you that normally every cellular process must find a way, or every cell, every DNA must find a way of repairing this error. So nobody is above mistake. That's what they normally say. The same thing with DNA. The same thing with every cell. The normal activities of the cell is prone to making errors, and the same way they are able to repair this error will actually depend on how perfect their repairing mechanism is. So if you actually want to maintain the fidelity and the integrity of the cell, that means there must be a very intelligent method of repairing DNA repair. So the DNA must make sure that it puts in place the mechanism, the correct mechanism and the right mechanism to be able to repair its error. And that's when we can now talk of the DNA is maintaining its integrity or is maintaining the fidelity. So of that, don't forget that integrity has to do with maintaining the normal composition. You maintain the normal composition. And when you talk of fidelity, they're actually talking about there's nothing like error of mismatch, nothing like deamination, nothing like. So all this can never be when the mechanism of repairing of the DNA is not being put in place by all the cell. So don't forget that the endogenous, what we have discussed so far, don't forget that we'll talk about endogenous sources of damage, and we look at how we have seen that DNA, we normally encounter some molecules of water, and when DNA encounter molecules of water, it will lead to a process called depurination. That is a loss of purines, and also the process of deamination. That is a removal or loss of either cytosine or uracil. Okay, and the second one is that DNA replication stress, the one that is due to stress. After replication, the cell does not take its time to go and rest. It's going to result into DNA replication stress, which is going to lead to incorrect or introduction of incorrect replication fork, and it's going to also lead to breaks and saturation. So having said this, don't forget that I've given you 3 tasks and which must be submitted today, just like you submitted yesterday. You are just being given to 4 hours from now, that is by 6:50. So you have to kindly submit that using the same mechanism that you use in submitting